that in the woody plants, it may be a gymnosperm or it may be in the dicotyledons, where they exhibit the woody developments or the secondary growth developments. There you can see two types of outer protective layers. If the plant at the primary growth stage, there you can see only the primary, the epidermal wall will be there, which is acting as a protective layer from the outside. Where when the plant reaches the secondary growth stage, I mean when the plant exhibiting the secondary growth, where you can see the epidermis is going to be vanished. I mean the epidermis will break down, and the function of protecting the internal structure of the plant body will be governed by the periderm itself. So the periderm will be considered as the outer protective layer after the plant which exhibiting the secondary growth stage. So the periderm, these are some of the anatomical details of a periderm drawn from the different plants. So there you can see, you cannot see that. So this is what from this to here. This is phellum, and this complete region is phellogen. Periderm broadly will go to the next slide. Will show that. So the outermost layer is the phellum. This is what the phellogen, which is falls below the phellum. And the next layer is the phelloderm. Usually the phellogen nothing but the cork cambium. So what happens here, what we are going to see the cambial cells. So when the plant at the stage of the secondary growth, in the plant can exhibit the secondary growth, nothing but when they are start initiating themselves for the secondary growth, what happens, the cork cambium will form. This particular cork cambium is going to divide periclinally. I think, but if single cell will divide periclinally, the outermost cell will divide, the topmost cell will divide and form the phellum, and the bottom cell will divide internal to the what we call the steel or the pith region, and those cells will be phelloderm. So all these three together, phellum, phellogen, and the phelloderm, all these three layers together will be considered as the periderm. Now let me take you to the characteristics feature of the periderm. So you already, as I already told you, that the epidermis and the periderm are the two outermost protective tissues in the plant body. Nothing but in the plant at the primary, when the plant is considered as the young which exhibiting only the primary growth, their epidermis, which is going to protect the internal structures of the plant body. Where when the plant reaches when the plant is about to begin, or when the plant is able to say that they are exhibiting the secondary growth, nothing but that they are forming the vascular cambium, cork cambium, then what happens, the periderm is going to form, and this periderm will be acting as the what we call outer protective layer. Nothing but these two which will protect the internal structure of the plant body during their particular time. So during the primary growth condition, there we can see the epidermis, and even the secondary growth condition, there the periderm is going to protect the internal parts of the plant body respectively. So what happens during the formation of a periderm, which is going to be initiated, or the triggering of the periderm initials are seen after the post-embryonic development. What happens, the meristem which will bring about the primary growth, that already we know that the meristems which will divide and form the apical meristem. For example, let us take the apical meristem. Here apical meristem will divide and form the shoot and the root. The meristems, when the plant body at the primary growth condition, these meristem will divide and are responsible for the formation of the shoot as well as root, leaves, and elongation of the stem and elongation of the root. I mean nothing but a primary body of the plant, or a primary growth will bring out the actual body of the plant body. I mean the actual body of the plant. Where there we cannot see any secondary growth, such as the what we call the cork or any woody structure. All we cannot see during the primary growth of the plant body. And the another one is once the plant is completing their primary growth condition, then it is shifting themselves to the secondary growth. Secondary growth, nothing but let us take, we have the intercalary meristem. So these intercalary meristems are responsible for increasing the girth or diameter of the stem. Similarly, there are some secondary growth, such as vascular cambium will come and it will start dividing and form the secondary xylem and the secondary phloem. So what happens when a stem which comprising this what we call this vascular cambium, it is going to release the secondary xylem towards this pith and the secondary phloem towards the periphery. Then what happens? It is going to increase the girth of a stem or a girth of a root. So such increasing in the primary structure of the plant body will be made by the primary growth, I mean the meristem cells, where the secondary conditions or secondary growth will be governed by the secondary structures of the plant body. It may be the vascular cambium, it may be the cork cambium. Right? Now when the plant reaches the secondary growth stage, we are going to see the secondary two or bifacial meristem. One is the vascular cambium, another one is the phellogen. Nothing but we already know that the procambium will result in the formation of primary xylem and a primary phloem, where a vascular cambium will result in the formation of secondary xylem as well as secondary phloem. Nothing but when the plant at the secondary growth driven which is been majorly controlled by the two meristem activity. One is the vascular cambium activity, another one is the phellogen. Vascular cambium activity, nothing but now the plant is at the secondary growth stage. The vascular cambium will divide and will form the secondary xylem and the secondary phloem at the center. And above that, we are going to see the phellogen, that is a cork cambium, and it is going to divide and form the phellum towards the periphery. I mean the what we call phelloderm toward the center. So then what happens? This particular cork cambium, or pardon, the vascular cambium and a cork cambium, they divide and increase this structure shape. What happens? We are going to see the girth or increase in the diameter of the plant bone. So here, how the development can be seen, nothing but how the vascular cambium and how this phellogen is going to form the different structures. So as again, I would like to repeat that in the vascular cambium, what happens? It divides. Let us take this. Is what let me what we call. You people have to imagine what I am explaining right now. Let us take now. We have a anatomical part of the plant body. Outermost is epidermis, cortex, and the steel. In the center one steel, and we are going to see the vascular bundle, and at the center we are going to see the pith. From epidermis, cortex, endodermis, I mean the steel, and the next is the pith region. Now the vascular cambium are going to form. What happens? The cortex gradually is going to be disappear. I mean the loosening of the cortex parenchymal cells will become, and the epidermis is going to be loose. Then what happens? The vascular cambium will form above the cambium. Think what? So we already know the procambium is there. It is forming the xylem, primary xylem, and the primary phloem, right? So above the what we call the procambium, what we are going to get? The vascular cambium, and this vascular cambium will result in the formation of the secondary phloem above and secondary xylem below. Nothing but what happens? This vascular cambium, to understand the position of a secondary and a primary phloem as well as xylem. What happens? Primary phloem, secondary phloem, cambium, secondary xylem, and primary xylem. Already the secondary vascular bundle, primary phloem, secondary phloem, cambium, secondary xylem, and the primary xylem. Such structure we are going to see. And what we call the secondary outermost phloem will be considered as the bast fiber or bast uncurricle. So what happens because of the extension of the secondary phloem? One more point, actually, Jyoti madam may explain at the time of explaining to the different anatomical type study. There are 1 second. Yeah. Phellogen. After this, above this, we are going to see the phellogen. Phellogen is again a cork cambium. Again, this particular cork cambium divides and forms the phellum, outerward division. Nothing but this and inward division. Nothing but this phellogen or a cork cambium will inward division leads to the formation of a phelloderm. This is cork parenchyma cells. Nothing but this cork cambium, above is cork, and next is cork parenchyma cell. So these 3 layers, and next we are going to see the 5 layers. Because of this particular big reason, we are going to see the increase in the thickness of a plant body. Overall, the phellum, phellogen, and a phelloderm all together will be said as the periderm or the outer bark. Not only a bark, it's been considered as outer bark. A complete bark. So how to say what is actually components of a bark is phellum, phellogen, phelloderm, and outer phloem. All these 4 components will be considered as a bark, where phellum, phellogen, and a phelloderm, these 3 will be considered as periderm or outer bark. Along with the phloem, if we consider the phloem also, then it is considered as completely a bark. Secondary growth stage, there you can see that this is the phellogen. This is nothing but cork cambium cells, and they divide and forming a phellum above. PS stands for phellum. And next we have phellogen. Again, internal division leads to the formation of a phelloderm. These 3 are the outermost layers. Nothing but just the formation of the phellogen and phelloderm and the phellum is been seen here. and you can see the cells of the cortex are again they obliterated. I mean, we can see that the cells are deteriorate. I mean, they are losing their strength and they're losing the capacity. Because of that, we can see there is an increasing in the what we call I mean, this was intercellular spaces. Apart from that, you can even see this brachysclerites, and you all can available fibers which are associated with the phloem, right? Nothing but the cork is going to lose its activity, and finally we are going to see that this complete region is going to be filled up by the what we call phloem, secondary phloem. And this is what the periderm. These three are the periderm, and this complete region after the typical development we can see outer complete nothing but outer layer, that is a complete typical bark. Is a typically of the functions of the periderm. Nothing but we already know that similar to epidermis, the periderm is also helps in protection of the internal parts of the plant body. So usually periderm, which is going to be formed usually, I mean, as consequently as per the biological clock of a plant body, I mean the phyto that what we phytological rhythms, the periderm is going to form. Apart from that, also during any wound, if there is any wounded or damaged condition in the epidermis, or because of the extensive growth of the plant body, there also we can see the complete new tissue is going to be formed, and that particular new tissue will be considered as the periderm. But the formation of a periderm mainly based on if the wounded region or the damaged portion of a stem as well as the root may come across the formation of a periderm. Apart from that, what we call extensive secondary growth, such as the phellogen formation and all those things. And the next is after all these developments, what happens? The epidermis is completely removed and a new complete tissue is going to be formed, and that particular tissue will be considered as the periderm. And this particular periderm, as we already know that the lentil cells are associated with the periderm, and they are helped in the exchange of water, direct exchange, and they provide a maximum support for exchange of a gases. It may be oxygen, it may be carbon dioxide, it may be water. Apart from that, this particular periderm is also having the water and the gas exchange part, but they contains the lentil cells here and there, and they helps in the exchange of a gaseous molecule because they lack the periderm lacks the stomata. As we already know that epidermis may have the stomata, but the periderm is nowhere having any such primary structure, that is a stomata. Because of that, the plant has to undergo the exchange of a gases. Nothing but the regulation of a gases has to be undertaken. So because of that, the periderm will allow the development of lentil cells. That's what we call it as a channel-like structures. And one more important function of a periderm is to protect the internal plant body from the pathogens, because we already know that as strong as periderm that you already come across, you may come across or you already come across in the pathology, that if the strong the periderm, if the strong is the epidermal region, then the infection is going to be very less. So the resistance to the different pathogen penetration is also been rendered by the periderm itself. So because of what happens in the periderm, because of excessive accumulation of a suberin, it is going to increase the thickness of a outer layer. There what happens, because of the increased thickness, it is going to become a very tough, and because of a tough reason, the particular what happens, the pathogen cannot penetrate, it cannot send its hostoria or any organs to infect the internal parts of the plant. So one of the best example we have in the potato. Again, the potato also may exhibit secondary growth. There it is showing a phellum layer comprising of many metabolites, a many layered phellum comprising of many metabolites, which helps in stopping off the what we call pathogen to enter the internal parts of the potato plant. And next one is the periderm in the grapevine, because we already know that lentil cells are seen only on the periderm region. Again, the lentil grapevine, the roots of a grapevine contains what we call, they are susceptible to the phellogen with the fungi. So because of many periderm layer in the root, we can stop the entry or we can make the grapevine root resistant to phellogen. And again, because of the periderm layer. And we have some of the what we call Norway spruces. Again, is the name of a plant. Again, ornamental plant. We can see it in India also. But Norway spruces are the woody roots, the plants where the woody roots of this particular plant are resistant to bud rot. So if you want to check, you just already have seen, and I think it in the image or in somewhere else, this is a cultivated plant. If you want, you can just Google it. You will come to know Norway spruce. What is that? No, Norway spruce. And because of the woody root content, woody roots, these particular Norway spruces are resistant to butter rot diseases. And now coming to the development of the periderm. I mean, how this periderm is going to develop and how it is going to form. We already know that epidermis, which is going to protect the plant body during the primary growth, where the periderm is going to protect the internal structure of the plant body during the secondary growth stage condition. Because epiderm is not functioning at all. So despite even we know that the importance of the periderm and also the what is the perfect performance of a periderm, the molecular mechanism which is underlying the formation of the periderm is not yet clearly elucidated by anyone, because we always put ourselves for understanding the secondary growth which has been formed by the vascular cambium, I mean the secondary xylem as well as the secondary phloem. The only concept is being focused from the many years, but the formation of a periderm because of the phellogen and the phelloderm and the phellum is not yet been clearly elucidated by anyone. We already know that morphologically we are going to see the 3 different layers on the bark. That's what outer bark, but the molecular underlying mechanism is not yet been clearly studied. But roughly many of the scientific community has put forth with the formation or development of the periderm, and that I'm going to explain in the coming slide. Okay, now, but the first periderm in the stem and the root, because we already know that the root and the stem both in both cases we are going to see the formation of a periderm, where in the stem the formation of a periderm is going to start immediately below the epidermis when the plant is about to begin its secondary growth. Nothing but at the initiation of the secondary growth stage, they what we call the below the epidermis only the periderm cells are going to be formed. Where in case of roots, the formation of a periderm is completely because of the pericycle itself. For the formation of a secondary growth, what happens once the secondary growth begins, the epidermal cells will get ruptured, where in case of in the root, the pericycle, nothing but pericycle, which is again the nearmost cell, nearmost tissue to the steel, nothing but above the pericycle you can see the cortex, hypodermis, and as well as the epidermis. What happens? This particular pericycle is going to develop and form the root phellogen. By what happens? The surrounding cells are going to be damaged because of the pericycle development, and finally it is going to wave off, or finally it is going to kill off, or finally it is going to slough off all the protective layer from the which are present above the pericycle as and when the pericycle develops, and finally it is going to form only the periderm, and that will explain in the coming slide. So can you all see Jones from the top? Phellum, phellogen, and a phelloderm. Right? So here they are taken from the internal. So phelloderm, the phellum, phelloderm, phellogen, and a phellum. That's what, for the easy understanding, they have made some correction. And let me tell you that this has been obtained from the current opinion in plant biology. Nothing but this is one of the top-notch journal. They are actually they are giving some information on the development or current opinions in the plant biology. From there only I have taken the image and some of the information. I'm just explaining in front of you from the again top-notch journal itself. Okay, now because this is complete, the formation or development of the periderm is not yet been clearly elucidated by anyone, and in most of the textbooks they are not given at all. Only the periderm they have given, only the lenticels they have given, and their function they have given, but they are not given the clear information about the structure, the development, and the formation. Okay, now so let us take this is what a mature wood. So this is what the periderm. There you can see the phloem, right, and at the center you are going to see the vascular cambium, and you are going to see the xylem. These are the, you can see there are large intracellular space with parenchyma cells. Formation of the periderm only in the roots of the Arabidopsis. Okay, now so what happens during in the conventionally and the plant at the primary growth stage, and they are about to reach the secondary stage, when they are about to start the secondary growth region, there you can see that the epidermis outermost layer, followed by the cortex, and followed by the endodermis, and at the next to the endodermis you are going to see the pericycle. Right. So what happens initially when the tissue and when the plant body reaches the signal, receives the signals, then the pericycle will start divide periclinally. Nothing but this is what is a cell, the pericycle cell, it divides periclinally. Nothing but outer cell and inner cell. Where this particular pericline division leads to the what happens? The endodermal cell becomes flattening, because only the division is appearing only at the pericycle region, because of increasing the number of cells, we are going to see the compression in the or the reduction in the flattening of the endodermal cell. These endodermal cells which are near to the phloem, because endodermal cell we are going to see the secondary phloem, where we can see the endodermal cell which are near to the secondary phloem, and they are going to, because of the pressure exerted, because of the continued growth in the periclinal division, as well as because of the outermost growth in the what we call outermost protective layer, other cells. What happens, these endodermal cell will get compressed and they undergo programmed cell death. Nothing but because of they don't have any other option, they are going to they will die. What happens, all the endodermal cells will not die as soon as the pericycle will start dividing. Where the pericycle will divide, nothing but this complete cycle, this complete pericycle patches is there, or only this particular region is showing the pericycle division, periclinal division, right? The remaining endodermal cells are will still there. Nothing but simultaneous pericycle division will occur. As the pericycle division increases, gradually endodermal cell will go and die. They undergo programmed cell death, nothing but apoptosis. So before undergoing the programmed cell death of endodermal cells, what happens, there are different markers. Nothing but there are different genes are going to be expressed by the endodermal cells, and they helps in the formation of a lateral root as well as the xylem differentiation. So just see here in the stage 3 or 4, here you can see that the the lateral roots length is more. So when you compare the lateral roots from the stage 1, stage 2 and stage 3, that in the stage 3 that you can see the lateral roots size is bigger, and it is the size of the lateral roots is still maintained in the stage 4 and stage 6 also. Can you before undergoing the apoptosis, it has released some of the markers or the genes or some of the phytohormones, so that what happens, the lateral roots are going to be increased their size, as well as the xylem differentiation can be seen in the lateral roots. Further, because after the death of after the apoptosis of endodermal cells, there we cannot see the further developments of the lateral roots, as well as the xylem differentiation cannot be seen. In the 3rd and 4th stage, again, what happens when the endodermis is complete, no longer present. When the complete endodermis is undergone apoptosis, what happens, the phellum, nothing but outermost cork cambium is going to be developed, and the phellum is outermost layer, it's going to be formed. This phellum is completely differentiated. So everything happening, what happens in the sequential order, consequently. So phellum has been already differentiated, it is going to be, again it has become suberized. So now what happens, the epidermis and the cortex are going to be broke down. So here, because of the pericycle division, there we are going to see the endodermal gradual apoptosis undergoing. Next, once the complete endodermis is gone or thrown out of the plant body, there what happens, where you can see the formation of the phellum, which has been already superized. So this is going to be seen in gradual, not completely. Here you can see that gradual apoptosis of endodermal cell, when in the 3rd and 4th stage, gradual removal of the cortex as well as epidermal cells. You can see there are the epidermal as well as cortex cells are not here, right? Then when the cell reaches the 5th and the 6th stage, at the 5th stage, what happens, the complete absence of the epidermis, endodermis can be seen. The 5th stage, the 3rd and 4th stage, still we have a few cells, and at the 5th stage we are not going to see any of the endodermis, only a limited or a negligible amount of the cells of the epidermal as well as the cortex cells can be seen. Finally, because of the periclinal tremendous activity, these what happens, the epidermis as well as the cortex cells will be thrown out of the plant body. Only at the 6th stage here you can see that complete a typical periderm. Nothing but overall summary of that particular development or formation of the periderm is that the cells which will undergo 2 types of the mechanism. One is PCD, nothing but programmed cell death. Once the cells have gone programmed cell death, they have to get removed of that. Abscission, nothing but the early death, because endodermis is not yet ready to die. Because of the pericycle division, the endodermis has to undergo the apoptosis. Even the epidermis as well as cortex is not yet ready to die. Because of the phellum, it has to go die. Similarly, programmed cell death as well as abscission are the two important independent mechanisms which are making the endodermis as well as cortex as well as epidermis to go uh, to broke down, and only the phelloderm is, or the periderm is going to form. This is what the complete developmental stages of the periderm. So here I would like to show some of the images, because further I don't have anything to explain right now. And can you all able to see the first image and the second image? So here, so this is what the wings of the China plant, again a landscape plant or ornamental plant, Eunomus alatus. There you can see that the formation of a corky layer. Can you all able to see this corky layer? So this is what the side view, and this is what a top view after this section. So just see the wings of this region and this region. Can you all able to see this complete region? Don't get confused with this particular center one. This region and this region, both regions have